Hello everyone. This is the first chapter of this course, and in this chapter, we'll learn about the way or methods by which we can establish a connection between a microbe and a disease. Which means, say, for example, now we know that Bacillus anthracis, this bacteria cause anthrax. Okay, so how the scientists established these things, that how they were able to say that this particular bacteria caused this particular disease. Okay, so so there are rules by which we do that. Okay, so whenever a new disease outbreak, a novel disease uh, is reported or outbreaks occurs, then scientists use some set of rules to establish the connection between that microbes and that associated disease. Okay, so so these rules were first proposed by a scientist named uh, Robert Koch. Okay, so this scientist, he proposed some rules which is also called postulates. Actually, these are called Koch postulates. Okay, and using this set of rules, we can establish the connection between a microbes and disease. Okay, so however, please keep it in mind that actually the original rules or postulates which was proposed by Koch has many limitations. So basically nowadays those original rules actually not used, rather some modified versions of the original postulates, or we can say that some alternative approaches are used to establish the connection. Okay, but the fact is also alternate approaches or rules are used. Basically these alternates rules have been proposed by other scientists to satisfy the original Koch postulates. That means although the original Koch postulates has limitations, so basically the modern versions or the modified versions or the alternate approaches that are used nowadays, those are basically to those have been basically proposed to meet or satisfy the original postulates in a alternate way. Okay, so anyway, we'll learn about all these things. So first, let's see the let's try to know about the original postulates. That means original Koch postulates. I think you all already know this Koch postulates, because I guess you have studied this in some earlier courses. Yet it's better to look again. Okay, so just for you can say just for recap, I'll just talk about the original Koch postulates first. Okay, so uh, Robert Koch, he proposed a set of rules by which we can establish a connection between a microbe and disease. So he proposed four rules. Okay, and these four rules are called Koch postulates. Okay, so here are this first, the first Koch postulate. It states that the microbes must be associated with the lesion of the disease. That is, the microbe should be present in all cases where symptoms of disease are evident and must be found in diseased tissues but not in healthy tissue. So in general, so if we want to say that this bacteria cause this disease, then this bacteria have to be present in all the diseased subject. Okay, and should not be present in a healthy person. Healthy person means who don't have that disease. Okay, so this is the first postulate. The second, now the second postulate. The second postulate directs that the microbes must be isolated from the lesions of the disease and grown as a pure culture. Okay, must be, we must have to isolate and culture the putative causative agents as a pure culture. Then the third postulate states that a pure culture of the isolated microbe should be reproduce the symptoms of the disease if it is inoculated into a susceptible host, either humans or experimental animals. That means to satisfy the third postulate, we have to inoculate or introduce the pure culture putative causative agent into a host. Okay, and this host can be a human or a experimental animal model. Okay, so if we introduce this pure culture microbes into that uh, susceptible host, then that susceptible host should get the same disease. Okay, so this is the third postulate, and in the first postulate it states that from this now from this diseased host we should have to re-isolate the same microbes as a pure culture. Okay, so these are the four cause postulates, and according to base this postulate, if we want to establish a connection between a microbe and a disease, then all these four set of rules or four postulates have to be proved or satisfied. Okay, now before the cause postulate, actually there are no such things by which scientists could establish this connection between a microbe and disease. Okay, so cause, so that's why actually cause postulates are very important, because he first showed that he first talked about the rules by which we can establish the connection. So still now the cause postulate actually regarded as a gold standard to establish this connection. Okay, I will actually add here that gold standard means the not the original cause postulate, because it has some limitations, but yet it shows the way. Okay, it actually shows the steps by which we can establish the connection. That's why it is still now regarded as a gold standard. Okay, so basically nowadays the modified versions of the cause postulates are used to establish the connection, but those modified versions are based on these original four postulates. Okay, so why these modifications was necessary? Why now we have to use the alternate approach to satisfy the original cause postulate? Because this original cause postulate has several limitations. Okay, so let's talk about those limitations. Okay, so the first limitations is based on the original cause postulates. The first postulates, it is assumed that the disease symptoms are dependent entirely on the bacteria. Okay, so why? Because the first postulate state that the bacteria should be present, or the putative pathogen should be present in a diseased host but should be absent in the healthy host. But so that means if the pathogen is present, the host should be disease. So which means, I mean, it implies that the disease is solely or completely or entirely dependent on the pathogen itself. But now we know that it is not true, right? Because the susceptibility of a host, that means whether he will get disease or not, depend on several factors, right? So for example, the genetics of that host and other factors like age, the natural immune system. Okay, so these things varies between host to host, like from one human to another human, or one animal to another animal. Okay, and the whether a pathogen will cause disease or not actually greatly affected by these other factors. Okay, so that's why sometimes even a healthy host is not diseased, even though they may have that particular pathogen in their body. Okay, so that's why the first, I mean, these are the limitations of the first postulate. Okay, so I mean, if we think about the real scenario, then we can see that the first postulate will not be true for every host. Okay, so that means even a a host have the pathogen, they may not have the disease. Okay, because in the original postulates, these other factors, the genetics and the age, natural immune system, these were not considered. Okay, and also another thing is we know that, I mean, like, so if a pathogen just get into a host, that host will not become sick, because every pathogen, I mean, there is a minimum number of pathogen which should enter into the host at a time, right? Which we called like ID50, infectious dose 50. Okay, so like suppose if one bacteria, say for example, one Bacillus anthracis enter into a host, the host will never get disease, right? A minimum number of pathogen should enter or infect the host to make the host sick. Okay, so these things were ignored or are not included or not considered in the first post-course postulates. Okay, so then another limitations, the second one. So the second course postulates assume that a pathogenic bacterium can be readily isolated and cultured. Okay, so it's very obvious that it's not possible for many cases, because actually till now we can culture only few number of bacteria or pathogens. Okay, so that means many pathogens, there is no system to culture many pathogens yet. Okay, the culture system for many pathogens is not yet available. Okay, so this was that's why it is the limitations. Then the third limitations. Okay, so the third course postulates assume that all member of a bacterial species are equally virulent, and that a single species cause each disease. Okay, so did really the third postulate assume that? Okay, so look back to the third postulate again. So the third postulate states that a pure culture of an isolated microbe should be should reproduce the symptoms of the disease if it is inoculated into the susceptible host, either humus or experimental animals. So what that means? So first we isolate, we have to isolate the putative causative agent from a disease host and have to uh, pure culture them. Then we have to inoculate this pure culture into a susceptible host, and that host get disease. So now think about the pure culture system. When we do pure culture, we basically try to uh, culture them as isolated colonies, and we know each colony in pure culture, each colony is originated from one single pathogen. Okay, so that means we can say that one colony, they actually consist of a single species. Okay, so now we know that all the I mean one colony is originated from a like a single strain. So now we know that all the strain of a bacteria, okay, they are not equally virulent. Okay, say for example E. coli, there are many different E. coli, right? And we know that and all the E. coli don't cause disease, and even all those E. coli who cause disease don't have the same kind of virulence factors. That means they don't cause same kind of disease. Okay, so these kind of things, I mean these detailed things is not considered in the original third course postulate. Okay, so that's why it's the limitations. Okay, now finally the final limitations. So original course postulates require that re-inoculation into a susceptible host to reproduce the disease symptoms. That means after doing the pure culture, we have to re-inoculate this pure culture into a susceptible host, and the host will get diseased. This is actually a important and big limitations. Why? Because there are ethical issues here. So for example, one human disease. Okay, so now in case of human disease, if we want to satisfy this fourth course postulate, then what we need, we have to inoculate the isolated pathogen into a human. It is unethical, right? We just cannot we just cannot inoculate a healthy host with a pathogen, right? it will be unethical. So this is that's why it is actually very big limitations. Okay, so although we can isolate the putative pathogen in a pure culture, we just cannot simply inoculate them into a healthy host. So now, to overcome this thing, what we can do? We can have to do some alternate. So I mean, we definitely cannot use the human. So we have to use a animal model instead of the human. So now this is actually a difficult thing. Okay, I mean, it may sounds very simple, but most of the time, I mean, in many cases, we know that diseases are kind of I mean, microbes are kind of host specific in many cases. Okay, or especially it is true for virus, but the same bacterium, they may infect different animals, but although they may infect different animals, they may cause disease into one animal and not in another animal. Okay, so it's actually really difficult to find a very good animal model to mimic the original human disease. Okay, so that's it's a limitation, because if we want to establish the connection, we need to satisfy the fourth ghost postulates. Okay, but as it's very difficult to find a host for a particular pathogens, it's actually also very difficult to satisfy this fourth postulate. Okay. Okay. So far we learned about the ghost postulates and their limitations. So now, I mean, in the next part, we'll learn about the limitations with some examples. Okay, we'll discuss kind of in more details about those about the ghost postulates and their limitations with examples. Okay, so it will be discussed in the second part, second video of this lecture. Okay, so thank you for your patience.